I want to briefly discuss the four levels of protein structure: primary, secondary, tertiary, and quaternary. It's important to understand these four levels as the structure of the protein determines how it behaves or functions in a particular environment. And this will be imperative to understand as we move our way through cell bio this semester. The four levels of protein structure are known as primary, secondary, tertiary, and quaternary. The primary structure is known as our linear sequence of amino acids. Secondary structure is determined by the hydrogen bonding between the backbone of the polypeptide chain. And we get two predominant conformations, either an alpha helix or a beta pleated sheet. The tertiary complex is the 3D dimensional shape that's due to the interactions amongst our groups. And then lastly, we have the quaternary structure, which is the result of the interaction between two or more polypeptide chains. So I'm going to go through each of these systematically and help you to understand them. So let's start with the primary structure. First of all, you should recognize what the basic amino acid looks like. Again, we've got our amino group, our alpha carbon, the carbonyl group, a hydrogen, and then the R group. And it's going to be important that you fully understand this structure. So when we are talking about a polypeptide chain, we're talking about the linking up of amino acids. And this linkage is going to take place between a carbonyl of one amino acid and an amino group of another amino acid. And due to a condensation reaction, we're going to have the loss of water and form this covalent linkage known as a peptide bond. So this is a very strong linkage. And when we're talking about the primary structure, it's really just what is the sequence of those amino acids. For instance, here we're looking at a serine that's next to an alanine, next to an alanine, etc. And some important features to notice on this primary structure is at one end we're going to end up with an amino group, so we call this our amino terminus, and on the other end of the polypeptide chain we have our carboxyl terminus. And you should be able to look at a structure such as this and identify where's the peptide bond. So for instance here, between this tyrosine and the glycine, we see the peptide bond between the carbonyl of one amino acid and the amine group of another amino acid. So this would be our peptide bond. Here's the next peptide bond, etc. You should be able to identify your alpha carbon as well as your R group. And the R group is what determines what particular amino acid you're looking at. So in this particular case, we're looking at a tyrosine. So again, that was our primary structure. And now we're going to talk about the secondary structure, which, as I already stated, is the hydrogen bonding between the backbone of the polypeptide chain, forming either an alpha helix or a beta pleated sheet. So now is when we're going to begin to see the actual folding of the protein taking place. So before we explore what it means to be an alpha helix or a beta pleated sheet, we should differentiate once again between what is the backbone versus a side chain. So a backbone is everything that's running along the backbone of the molecule, not the R groups. So the R groups would be considered our side chains. So again, the amino group, the alpha carbon with that hydrogen and the carbonyl, that all would constitute our backbone. And so right now we are not talking about any of the interactions happening between the side chains or our R groups. So when we're talking about secondary structure, again, it's that alpha helix or beta pleated sheet. It's going to be due to the interaction between an oxygen of a carbonyl group and the hydrogen that's coming off of an amine group. So let's first of all look at the alpha helix as shown here. This is going to be a coiled structure that again is due to hydrogen bonding between the backbone of the molecule. So between a carbonyl in the backbone and the hydrogen that is bound to an amine group. So you can see that taking place here. The side chains do not play a role in this hydrogen bonding. They're oriented outward away from the helix, so they do not confer this particular type of folding. Now, a beta pleated sheet, on the other hand, is due to hydrogen bonding taking place between parallel sections of the backbone chain. So what you can see here is we've got the oxygen of a carbonyl group, again interacting with the hydrogen of an amine group, but this is taking place between parallel sections of the polypeptide chain. Again, an alpha helix is going to be due to the hydrogen bonding taking place between adjacent amino acids. So actually every 4 amino acids you can see this interaction between the oxygen and the hydrogen, whereas here we're talking about parallel sections of the backbone chain. So far we have talked about primary structure, secondary structure, and now we're going to talk about the tertiary structure. So the tertiary structure is what gives it a particular 3-dimensional shape, and this is due to interactions that are taking place between the R groups. So now we will begin to talk about those actual R groups. So between the R groups we can have lots of different interactions that can take place, and this is going to cause the protein to undergo a particular folding. And this is going to be incredibly important for the structure of the protein, because all of a sudden you can bring together amino acids that were really far apart from each other. You can bring them very close to one another. So let's talk about the 5 different types of bonds that confer this particular folding of a polypeptide chain. So the first one I want to talk about is the hydrogen bonding. So now this is hydrogen bonding that's taking place between the R groups. So that's nicely shown here. We've got these different R groups that are extending out from the polypeptide chain, and now you can see we've got an oxygen that's interacting with a hydrogen that's a part of this R group here. Secondly, let's talk about the ionic bonds. So ionic bonds, again, are going to be the interaction between a positively and negatively charged amino acid. And so we're going to be looking at our acidic and basic residues here, those R groups that are going to be interacting with each other to get the ionic interactions. Third, we've got the hydrophobic effects. And so again, hydrophobic is a fear of water. So that's going to be our non-polar amino acids. And in an aqueous environment, so if you imagine within the environment of a cell, so in the cytosol, that's an aqueous environment, all those side chains that are non-polar and are hydrophobic are going to want to flee from the water. So what we oftentimes will see is that those non-polar amino acids are going to be clustered on the interior of the protein. And those non-polar amino acids are going to want to interact with each other. If you think about like dissolves like, again, though, you'll find them oftentimes clustered on the interior of a protein. Fourth, I want to mention van der Waals interactions. These are going to be very weak interactions, and these are due to attractive forces that take place between atoms. And this is going to be true for all 20 different amino acids. There's going to be this attractive force that is taking place, like a magnetic force. And these, again, are very weak, but collectively they're going to have an impact on how the protein folds. And lastly, I want to mention the impact on the folding of a protein due to disulfide bridges. So this is going to actually be a covalent bond that's going to form between 2 cysteine side chains. So if you go back and look at all the different 20 amino acids, you can see what a cysteine looks like. And again, the R group here is just a CH2 bound to a sulfur, and these 2 sulfurs can actually form a covalent linkage. So this is actually a very strong bond that can bring together really distant parts of the protein together. All right, so we have now covered primary, secondary, tertiary structure, and last we have quaternary structure. And what quaternary structure is again the interaction between 2 or more polypeptide chains. And so we've gone through our primary structure, secondary, which shown here is an alpha helix or could be a beta pleated sheet. We've got our tertiary structures. And so then when you bring together your individual polypeptide chain and allow for an interaction with other polypeptide chains, we're going to get this quaternary structure. And the types of interactions that different polypeptide chains can have are the same as what we found with our tertiary structures. So you can have hydrogen bonding taking place, you could have van der Waals interactions, some of those ionic interactions, hydrophobic effects, and even the formation of disulfide bridges. So hopefully you feel more confident in being able to explain the 4 levels of protein structure. I would highly recommend that you go through this final slide with some of your colleagues and try to explain in your own words the 4 different levels of structure. And again, why are we covering this? Because structure determines function. Proteins are very dynamic in nature with regions that can move and bend, and the weak bonds, like we talked about, such as hydrogen bonding or van der Waals forces, they can confer a particular folding on the protein, even though they're weak. But because they're weak, they can come apart and then they can reform. And so this large number of bonds within the polypeptide chain are all responsible for conferring the structural integrity of a particular protein. Thanks.